This lecture is called Cellular Respiration. It is about cells and how they use glucose and oxygen to make a lot of ATP in some circumstances. It would be helpful if you would follow along with the notes that you've been provided on this topic right here. First of all, you'll see this equation that looks kind of familiar but maybe a little bit different in some ways. And if you look carefully, it's essentially the same equation as photosynthesis but backwards. And what we have going on is glucose, C6H12O6, and oxygen combine to yield CO2, water, and ATP in the form of energy. And what this represents is how we and all other life on the planet, all life, protists, fungi, plants, animals, bacteria, everybody, how we get ATP out of the foods that we consume. Now, we all know that glucose is a sugar, and we don't just eat sugars, although some of you may like to just eat sugars. But we're going to use this as our model for understanding how cellular respiration works. Now, this whole idea of respiration sounds like breathing, and now you look carefully and you'll see that, well, oxygen is a gas and carbon dioxide is also a gas, so it kind of is like breathing. This schematic here shows you how glucose, as our reactant, will go through a couple of stages to become a pyruvate molecule and then either a waste product of alcohol, ethyl alcohol, and some CO2, or lactate, or it will enter mitochondrion, plural would be mitochondria, to create a lot of ATP. And we'll talk about the different circumstances under which these would happen. So let's talk about the stages of respiration. But before we do, take a look at this mitochondrion. They are double layered in terms of their membranes. They have two membranes. You can see the outer one is smooth, while the inner membrane has lots of these folds. These folds are called cristae, and inside the cristae, the very inner part of the mitochondrion is called the matrix. You can relate it to the stroma of the chloroplast. First stage of respiration is glycolysis, and the second one is the Krebs cycle, otherwise known as the citric acid cycle, and then third is the electron transport chain. Your book mentions two stages, but it doesn't separate these two out, and I'm going to separate them out because they have such unique processes, and it's best if you remember them separately. Now, glycolysis, we say, is anaerobic because it does not require oxygen, whereas basically the electron transport chain does require oxygen in the very, very end, so it is aerobic. And we don't even get to the Krebs cycle unless oxygen is present, so you can basically say that it's aerobic as well. So let's start with glycolysis. Now, all life on the planet does a glycolysis. All life. Even those bacteria that live at the deep sea vents way, way at the bottom of the ocean. And don't get freaked out by this series of reactions over here. We're just going to highlight a few of the parts. First thing you need to know is that this happens in the cytoplasm. Now, you should remember from way back in 7th grade that the cytoplasm is basically the goop in between the cell membrane and the nucleus, if a cell has a nucleus. So it's just in the abyss of the cell, not inside an organelle. Now, this is important because bacteria, you might recall, are prokaryotes. They do not even have organelles, and they do cellular respiration too. So, of course, glycolysis has to occur just in the cytoplasm. Now, this process, as I mentioned before, does not require oxygen, therefore we call it anaerobic. And let's look at the details that you need to know. Over here it looks intimidating, but there's not that much you need to know. First of all, glucose, you know, is a 6-carbon molecule, and it gets split into two 3-carbon compounds. And these 3-carbon compounds will eventually get turned into 2 pyruvates. Pyruvates are also 3 carbons. Now, this process requires 2 ATP. We have to spend it, okay? We have to invest some ATP. In the end, though, we make 4 ATP. Notice the direction of the arrows. And we also make 2 NADH molecules, which is similar to the NADPH that we talked about in photosynthesis. Has the same role, but it is a slightly different name. Notice then that we're gaining a profit of 2 ATP. We had to spend 2, and we earned 2, so we really took home 2 ATP. Keep that number in mind for later on. This pyruvic acid, let me back up a second. So remember we made pyruvate or pyruvic acid. There are two possible fates of this. This can either go one way or the other, and it all depends on whether or not oxygen is present. So first of all, if it goes into the Krebs cycle, then there would have to be oxygen because this is an aerobic process. This happens in the mitochondrion itself, and we get a lot more ATP this way than the other way that we'll talk about. We'll get up to 38 ATP if we go the aerobic route. Fermentation is the other route. If there is no oxygen available, in other words, an anaerobic situation, then we would go into fermentation. Now this again happens in the cytoplasm right after glycolysis, so there would be no mitochondrion involved at all. And if you're a bacterium, you don't even have a mitochondrion, so nothing to worry about. This does not produce any more ATP, so note again, fermentation does not produce any additional ATP, but instead it regenerates a molecule called NAD+. And this would then allow glycolysis to continue. And remember, glycolysis did make 2 ATP, which is better than nothing. So fermentation, please know that this is not part of aerobic respiration. It is not. Let's talk about fermentation for a moment. There are 2 types of fermentation. First, there's alcoholic fermentation, or alcohol fermentation, and this is done by yeast cells. Yeast are single-celled fungi, which is kind of interesting. And with them, you have that glucose, it goes through glycolysis, and it generates the pyruvate molecule. The pyruvate then go through this intermediate 2-carbon compound, but in the end make ethanol, which is a type of alcohol, and they release CO2, which as you know is a gas. Now think about bread rising and yeast, right? If you're making bread, you need to add yeast to it. And that's the whole reason it rises, is because of them tooting out CO2. And as I've mentioned before, those little holes that you see in bread, those are basically the bubbles of CO2 that kind of remained in that shape. But the whole point of this is not to make the alcohol or the CO2, which is interesting, but instead the NADH then gets converted back to NAD+, which is a reactant here. We need NAD+ in order for glycolysis to proceed. So the whole point is to recycle NAD+. Now, in the event if you are making beer or wine, then this end product is, of course, the ethanol is interesting to you because that's the whole point of making alcoholic beverages. The other type of fermentation is lactic acid fermentation, which occurs in animal muscle cells, including our own. And pyruvic acid, in this case, would be converted into lactate. Thank goodness we don't make CO2, because I don't think bubbles of CO2 coming out of our muscles would feel very good. But this happens to us when our muscles basically use up oxygen too quickly or faster than we need more than we can get. And so our bodies will switch from aerobic respiration to anaerobic respiration, and we will do lactic acid fermentation instead. And as you've read about, it used to be thought that lactic acid or lactate created basically inflammation or irritation to your muscles, but it's been more recently refuted and found that your muscle soreness after a very intense workout is actually just due to micro tears of your muscles, which will repair themselves over time. But it's still good to cool down anyway from a hard workout to digest some other metabolites. And this is just an area where probably beer is being made in large vats. Now, what if oxygen is present? Then what? Then we're going to enter what's called the Krebs cycle, which is also known as the citric acid cycle. Citric acid I'll show you on the next slide. It's basically right over here. Krebs is the name of the scientist that first described it. Now, he didn't work alone. One of my college professors actually worked with Krebs, which was pretty amazing. As you proceed in your biology career, you'll come to appreciate how important Krebs cycle is. And so for me to have had a college professor that worked with Krebs was pretty impressive. What you should know about the Krebs cycle is that it happens in the mitochondria, and it does require oxygen, so we would consider this to be an aerobic process. To go through the steps, I'm going to use a different figure that I like a little better because it actually gives names to a couple of these compounds that gives it some identity instead of just using all these numbers. So first, this is the figure I'm going to use. Now, before we even get into Krebs, by the way, remember we made pyruvic acid during glycolysis? That was out in the cytoplasm, or in the cytosol, before we ever even came into the mitochondrion. So before we get into Krebs, that pyruvic acid gives off a CO2 and becomes acetyl-CoA. So acetyl-CoA is what actually enters the Krebs cycle in the mitochondrion. Let's consider the details that I'd like you to be aware of. First of all, that acetyl-CoA will combine with oxaloacetic acid. You know you want to say that 5 times fast. Oxaloacetic acid. These then form citric acid. So that's where the name citric acid cycle comes from. And this is a 6-carbon compound, which makes sense because acetyl-CoA is 2, oxaloacetic acid is 4. So here we have citric acid. Then, through these steps, I'll just kind of summarize. Here's what you need to know. We lose some carbons. We lose some carbons here in the form of CO2. There's some gas. And then here's some CO2 gas. Now think about this. CO2 is a waste product that we want to get rid of. And when you look at the cellular respiration equation, it is a waste product. So when our tissues are doing Krebs and the CO2 is coming off, those will then attach to the hemoglobin in our red blood cells. Those red blood cells will work their way back to our lungs, and then out they go, out your trachea, out your mouth and your nose, and that's what you're respiring. So it really came from way inside of your cells. I think that's pretty amazing. Also, what we produce in steps 2 and 3 is some NADH here and here. Now those are going to be important later on. They serve the same purpose as NADPH as we talked about in photosynthesis. Next. Steps 4 and 5. So we're just going to go right through these guys right over here. This 4-carbon compound that was generated, and you don't need to know those at all, is going to be recycled back to oxaloacetic acid. Because it's a cycle, we need to keep going. But along the way, notice we're producing FADH2 and NADH. So pay attention that we have made 1, 2, 3 NADHs, excuse me, there it is, and 1 FADH2. That's going to be important. So these NADHs and FADH2s are then sent to the electron transport chain, which should sound familiar because we talked about that one in photosynthesis and how that is where we made a lot of ATP during the light reactions, which then drove the Calvin cycle. But here in the cellular respiration, it's backwards, where we are going to make our NADH and FADH2s first, and then go to the electron transport chain so we can make a bunch of ATP, because that's the whole point, is to make a bunch of ATP. Alright, so let's go to that electron transport chain. This is a better image than the one that we saw before with photosynthesis. Here you've got your phospholipids, these little guys with the legs. We'll talk more about those in our next unit. Here are a series of carrier proteins through which the electrons get transferred. And let's talk about what's happening there. Oh yeah, and here's my favorite enzyme, you might remember, ATP synthase. So you need to know where the electron transport chain happens. That happens on the inner membrane of the mitochondria, those cristae. So those NADHs, remember those right here? And this picture unfortunately does not show you FADH2. I'll explain that in a second. NADH and FADH2 from glycolysis and Krebs, remember we did make some NADH way back in glycolysis, they go all the way in through the cytoplasm and into the mitochondria and are used right here to drive the electron transport chain. So the role of these guys is to carry high energy electrons from glucose. So it's all about the electrons, the negative charge. It's electricity that makes the whole thing work. It's really pretty interesting. So the energy from the electrons, so remember these are electrons, and instead of getting zapped by photons of light, that's not going to happen. They're just stripped off of these molecules and they're sent down the electron transport chain. But when they're doing that, we're using this energy to pump protons out. So we're taking these protons, these hydrogen ions that have lost their electrons, and we're pumping them out into this little space in between the inner membrane and the outer membrane. So we're creating a gradient, a concentration gradient across the inner membrane. Now these protons want to diffuse back in to equal out the hydrogen ion balance. So what will happen, I guess I should go to the next slide, is the hydrogen ions will then diffuse back through ATP synthase And remember that rotary head, it spins, that I talked to you about. Right in here, ADP and a phosphate will then combine to form ATP. So just that flushing of the hydrogen ions through ATP synthase will create a bunch of ATP. So why do we get so much ATP? We make a lot. 36 to 38 ATP are produced for every molecule of glucose. So for every C6H12O6, we get this much ATP, which is a lot. And here's why you get so much. See this NADH right here? Notice that the electron goes through 1, 2, 3 carrier proteins. And at every one, you pump out 1, 2, 3 hydrogen ions. And that hydrogen ion is eventually going to come back through ATP synthase. And for every hydrogen ion or proton that comes through, you're making 1 ATP. So NADH enters right here, and you get 1, 2, 3 hydrogen ions, therefore 3 ATP. What's not shown in this picture is FADH2 will enter right here on the second carrier protein. So it only activates 2 carrier proteins. It probably has to do with the shape of FADH2 and the shape of these proteins here. But regardless, for every FADH2 molecule, you get 2 ATP. So if you count back to how many we made to begin with, you'll see it all adds up. Now at the final end here, you've got this proton that came in, and it's got to go somewhere. Otherwise, we're going to have this accumulation of protons, and it's all going to back up. So at the end, this hydrogen ion will actually combine with water. It'll combine with oxygen and electrons to make water. I should be looking at this hydrogen ion. So here, the hydrogen ions will then combine with oxygen, protons, and electrons to make water. So your final electron acceptor is oxygen, and that's very important. And then we'll make water. Without oxygen, it all backs up. There's nowhere for these electrons to go, and then fermentation will happen instead. So the point here is once we've gone through glycolysis, which does not require oxygen, happens out in the cytoplasm, you net a whole 2 ATP. Without oxygen, you're only going to make about 2 ATP. With oxygen, you're going to make a bunch, up to 38 ATP. So we are much, much more efficient in making our ATP in the presence of oxygen. Again, about 38 ATP in aerobic respiration versus just the 2 in anaerobic. And remember, the 2 actually came from glycolysis, not from fermentation itself. This slide here is just to demonstrate and remind you that all foods are actually broken down into their building blocks. We eat proteins and carbohydrates and fats, which are your lipids, and of course nucleic acids are in everything that we eat. And all these building blocks are going to be processed through cellular respiration. But because they have different shapes and makeups, they're going to enter the cycle at different stages, which isn't a big deal. And we take sugars or glucose because they start at the top and go all the way through. And you don't have to know where glycerol or fatty acids or amino acids join, but just to show you that they all do get processed through cellular respiration, they just enter at different places. So I hope this was helpful.